Hello, I am Frank Alain and I'm a biology and chemistry teacher at IB at Bergen Katedralskole in Bergen, Norway. And I'm going to go through high level paper 1, May 2016 exam. And we start with the first question. Question 1. It says, which molecule is a sugar? And of course, this pentose is a sugar. And the way you can see that something is sugars is the hydroxyl group. Sugars are basically carbon skeletons with a lot of hydroxyl or OH groups. Here we can see B is a lipid and C is an amino acid with an interchangeable R group. And D again is a fatty acid. So D is the same as B. B might be a steroid hormone or a cholesterol or something, but D is a fatty acid. Here again the acid group. Number 2. Why is sweat a good coolant for the body? And the correct answer here is B. Because water molecules, if I draw 2 of them here, they have bonds, hydrogen bonds between molecules. So from this hydrogen, which is a little bit positive, there is a hydrogen bond to this oxygen, which is a little negative. And if this evaporates, this bond is broken and you get kind of 2 free oxygen without this bond. And to break that bond, you need energy, and that energy is heat taken from your skin. And that's why sweat is such a good coolant, because it contains water. Which molecule is a polysaccharide? And the answer is, well, glucose is a monosaccharide, so it's not that. Glycerol is a component in fat, so no. And glycogen is, of course, a polysaccharide, because it is it looks about something like this, and it's found in the liver, and it's kind of the animal version of starch, and we have it in the liver, and from this we can mobilize the monomers, which are of course glucose. Glucagon is a hormone released from the pancreas to actually make this happen. So, but it's not a polysaccharide itself. It's actually a peptide hormone. Number 4. Which substance is used for structure in plants? So amylopectin is part of starch, and collagen is used for structure but in animals. Starch is what amylopectin and amylose make, but it's still storage. So it's of course cellulose. Cellulose looks a little bit like starch or glycogen, but it has a completely different bond. And also the sugar molecules are not alpha glucose as in starch, but beta glucose. So the components are a little bit different, and the bond between the monomers are very different. 5. What usually distinguishes DNA from RNA? And A, strands are symmetrical. No. The DNA code is not symmetrical, meaning the order of the bases cannot be read the same way if you read from the 5' or 3' end. There are palindrome sequences, but they are short and rare. Okay, B. Contains adenine? Well, yes, but the existence of adenine in DNA and cytosine in RNA does not distinguish. So this is an example where the what is claimed is actually not untrue, but it's still irrelevant compared to the question. And pentoses linked to phosphate and pentoses linked to bases. Okay, that's also true in itself, but it doesn't distinguish between DNA and RNA. So it's of course D that is correct. Double-stranded, that is DNA, and single-stranded, that's RNA. 6. Soon after fertilization, parental epigenetic methylation is reversed in DNA, okay? And then later, tissue-specific epigenetic modifications are made to the embryonic DNA. And here, this graph shows the degree of methylation. So, methylation is something that happens to DNA. One of the bases gets this methyl group, which looks like this. It's just put on instead of a hydrogen. And that signals very often to the body that don't make this protein. But you see that after fertilization, like the methylation that was present in the parents are removed, and then you don't get it back until you have embryonic DNA. And I think this is because the embryo starts making tissues, and methylation is tissue specific. So it makes no sense to have kind of any arbitrary methylation in the blastocyte or something which don't have tissues yet. So this kind of makes sense for me. But let's look at the question. So it says, what are the changes in DNA methylation during embryonic development? Only the parental DNA becomes demethylation. Only the paternal DNA becomes demethylated, and that's untrue because we see here the maternal. We see the key here. This is the maternal. The maternal DNA is also demethylated. The maternal DNA becomes demethylated first. No, we see that that's the father. So father first. Okay. See, the methylation patterns of the parent's DNA are erased before fertilization. Well, fertilization actually happens when you make the zygote, but the demethylation is complete long after starting the embryonic development, actually after the embryo has formed. So meth. So this is also wrong. So we have we can cross out the 3 first as wrong, and then of course the last one must be correct. But why? The methylation patterns of both parents are erased after fertilization, and yes, that is true because we see here they are erased, and that is indeed after fertilization. So of course reading the label of the y-axis here is essential, and all of these absurd complicated cases is not actually, it's not expected that you actually know this detail about methylation, DNA methylation, epigenetic methylation in embryonic development. It's just expected that you can understand the graph, and that you should be able to do. So when you get these questions, don't let yourself be confused by the heavy theory, because it will be there. It will be there to confuse you. You have to kind of put your glasses on, tunnel vision on, and just look at the axes, and yeah. So the y-axis, the x-axis, and then look closely at each alternative. So 7, which sequence of DNA would be suitable in DNA profiling? And for DNA profiling, the whole point is that you kind of cut the DNA strand like this. So now I'm just showing with a single strand. Of course it would be double-stranded, and this would be, so you would maybe cut to the other strand here. But for these, so these cuts would preferentially make unique segments. But to have unique segments, you need a unique sequence. And in this question, actually D is the correct one. Why, you might think? Well, let me just show you all the different bases in A. So we see that here in sequence A, B, C, it's just a C, but in D it's an A. It's an A, and in here the second last base is also changed here, but also in C, and then in the third last it's changed here, and the fourth last is changed. Let me see here. Here, here, and here. The second last is here and here and here, and the third last is similar in all, and then this one is just changed in C and D. Maybe it would be correct to move this one here, because here it's actually and let me see, yes, and then yeah, then it's similar, and we see that clearly. The D sequence has most differences compared to the other. Let me just check. Yes, it was. I just checked the mark scheme. It was D that was different. So this is 1, 2, 3, 4, 5, 6, but this is only 1, 2, 3, 4, 5. 8. The image represents a model of transthyretin, and it asks structure X, which level is it? And as you might know, the primary sequence in a protein is considered the sequence of amino acids. For example, lysine, alanine, methionine, and so on. Okay, that is kind of primary sequence. Secondary sequence is when these amino acids make some kind of universal structure, like you have something called alpha helix and a beta sheet, like this alpha helix is like a thick coil, and that is indeed what is shown here. This is a alpha helix, and maybe, I'm not sure, but I think this is a beta sheet. Okay. A tertiary structure is what this black makes. This black makes some stuff, and quaternary is when you take different peptides, like you take the black one, the white one, and you see the gray one behind here. When you take them all together and make what this might be a 7 helix membrane. It looks like there is an opening here, you see, and then it looks like this is inserted in a membrane. I haven't checked transthyretin and Wikipedia, but it looks to me that you have a lot of hydrophobic amino acids here, and that this is some kind of channel, maybe a 7 helix membrane protein or something. This is just me speculating. Don't arrest me if I'm wrong. So B here is correct. 9. In one of the curves in the graph, the rate of enzyme catalyzed reaction has been plotted against the substrate concentration. So plotted against the substrate concentration, this is very important information. So this is typically enzyme activity. And C would of course be a non-competitive inhibitor. Non-competitive. D, like there is no enzyme activity. Maybe this is just you have added something that just cuts the enzyme into pieces or something. I don't know. But A is kind of, that is probably the uninhibited. And of course D is the competitively inhibited. So small quantity, and it's a small quantity of the inhibitor, and it's competitive. So what does this actually mean? Well, let's say the enzyme is a Pac-Man, and this mouth is the active site. Then the substrate goes into the mouth, but then the inhibitor maybe looks very much like the substrate, but it has maybe a what's called a thorn or something that makes it stick in there and prevents these blue ones, the substrate, to get to it. So since these two are competing for the same space, if there are many more blue ones, they will kind of outcompete the few red ones, okay? So we see that with very high substrate concentration, so these blue ones in my analogy is the substrate, the red ones are so far in between that they can't actually have a meaningful impact on the enzyme activity. But on the other hand, if there is very little substrate but relatively more inhibitor, the inhibitor has a great effect. If it's a non-competitive inhibitor, then the site for the inhibitor is actually not the active site, but it's kind of a control site, and the inhibitor would have access to this site independently of substrate concentration. That's why you see no effect on the substrate according to substrate. So see here, it's constant after a short while. Number 10. The diagram represents the Krebs cycle, which processes are occurring at 1 and 2. So let's just look at 1 first. And here we see that NAD plus is reduced to NADH plus H plus. Usually when I teach this, I say that NAD is kind of a lorry, and it's empty if it's NAD plus. But, so empty, NAD plus, okay? But if it's filled, then it's NADH plus H plus. And then it has a protein and an electron. Okay, so NAD plus if it's filled. And this process of giving something an electron, if you have chemistry, you know very well this is reduction. If you don't, just remember, if you get an electron, you are reduced. If you lose it, you're oxidized. It's because oxygen always takes the electron. Here, we have two things going on. We have a decarboxylation. Decarboxylation. When see the C5 compound here is becoming a C4 since one carbon dioxide is going out. But then also we have this reduction of NAD plus, meaning then of course the C5 has to be oxidized. So let's look at this. So in 1, C4 is being reduced. No, this is wrong because of course it was NAD plus that was reduced. B, NAD plus in 1 is being decarboxylated. No, this is happening to the carbon compounds. NAD plus is just the carrier, and it's never actually changed other than being reduced or oxidized. C, NADH is being reduced in 1, and yes, we saw that. And C5 is being decarboxylated. Yes, C5 became C4 and lost the carbon dioxide. So just control with E. It says that NAD plus is being reduced. Yes, it was. And in process 2, C5 is being decarboxylated. So yeah, so here I actually said wrong, so sorry about this. So NADH is of course not being reduced in process 1. If you look here in process 1, actually NADH is formed. So no, there is no reduction of NADH. Sorry, it's the NAD plus that is reduced, okay? Because it's NAD plus that gets the electron. Sorry for my confusion. So it's D that is the correct answer here. 11. Which gas produces most of the bubbles in bread dough? And in bread dough, there are two things you need to know about the dough that is rising. So you put it to rise before you put it in the oven. And one thing is that you use yeast, you add yeast to it like this. It's kind of like a protozoa slash, yeah. No, sorry, of course not the protozoa. Yeast are classified in the fungus kingdom, but they are eukaryotic. Okay, so you put the yeast into the bread, and so that's one thing. So you have a microorganism that have access to fermentation, like it can have aerobic processes, respiration, but it can also have anaerobic respiration, meaning fermentation. The other thing you have to know is that oxygen doesn't like penetrate very easily into the dough. So indeed there is more fermentation, or at least like the yeast takes all the oxygen it can, but then the rest is done by fermentation. So water vapor is only used during aerobic. Only aerobic, but carbon dioxide is made during both aerobic plus anaerobic. And since there is some oxygen diffusing into the dough, and of course that aerobic respiration is more efficient, but since it's both aerobic plus anaerobic, and since both of these processes create CO2, but only one of them make water vapor, then of course C is produced in most of the bubbles in the bread dough. And what are these bubbles? Well, the bubbles is actually what makes the dough rise. 12. Which molecule would first contain carbon-14 in the algae chlorella if it was grown in the presence of light and radioactive carbon dioxide? So here we are growing a green chlorella algae, and so we have water inside here, and then we have carbon dioxide maybe in the air, and of course dissolved into the water. And this carbon dioxide is not C12, the normal, it's C14, a radioactive form of carbon. And then probably, like the setting here probably tells you that it was first grown in normal carbon dioxide, and then you change the carbon dioxide source. So and then it's grown in the presence of light, so photosynthesis occurring. So the light reaction splits water and makes some NADPH plus H plus and so on, and all this fuels the Calvin cycle. Calvin cycle. Okay. And all of these components here are from the Calvin cycle. So let's see if I can remember. So if I remember correctly, ribulose bisphosphate, RUBP, is what is used, is the kind of one of the substrate for Ribisco. So Ribisco is a very important enzyme. It's kind of the enzyme that take inorganic carbon, for example, inorganic carbon in the form of carbon dioxide, and makes it organic carbon. So Ribisco represents the frontier between the living and the dead in our world, or the organic and the inorganic. So it takes one ribulose bisphosphate and then adds on a carbon dioxide, and then you get a glycerate 3-phosphate. So I just say glycerate 3-phosphate, and then I just draw the phosphate like this, okay? And then this again makes ribulose it makes, what is it called? I don't remember. It's triose phosphate. Haha, I remembered. Triose phosphate. Okay, so it's another 3 carbon. And then this makes, like if you have 1 triose phosphate, it makes half a glucose. Or if you have 2 triose phosphates, you get a whole glucose. So GLC, that's glucose. But not all of them are taken out. Some of them are used to make more ribulose bisphosphate. So, sorry for the long explanation, but the point is that we see very clearly that the first, like if we suddenly change from normal carbon dioxide to radioactive, we see that the first compound it's included in is the glycerate 3-phosphate, and then the triose phosphate, and then ribisco. And then when you have made a lot of sugar, and that sugar is being used to make amino acids, and those amino acids he used to make proteins like ribisco, then finally ribisco gets the radioactive carbon last of these 4 alternatives. So the correct answer here is glycerate 3-phosphate. 13. What is the process shown in this image? And we can see very fast that this must be a prokaryote. And the reason is that there are no organelles, no nucleus, and we see sign of a cell wall or even capsule. So the correct alternative here is binary fission, because these other alternatives refers to it as a eukaryote, and C refers to a process, mitosis, which involves nucleus, and that's only in eukaryotes. So mitosis is a eukaryotic process, so that's also wrong. 14. What can be deduced about the striated muscle fiber from both of these statements? Eukaryotic cells has 1 nucleus, striated muscle fiber has many nuclei, and we see that this is an exception to cell theory. So cell theory is kind of a basic rule that have some famous exceptions. Like a eukaryotic cell has only 1 nucleus. Yes, that's usually true, but in some hyphae, which don't have kind of sections between the segments, you can have many nuclei. But since if we talk about striated muscle fibers there, that is the correct one here. Even though C is also refers to an exception to cell theory, B is more correct. 15. The diagram is a model of a type of movement across the membrane. And here we have a molecule with high concentration, because there are many. You see that there are many molecules here. And so it goes to low concentration. And so this is movement along its concentration gradient, and this is of course facilitated diffusion. And it's not simple, because then these molecules would just go over the membrane by itself, which is very slow, because this molecule is probably a little polar, and then it wouldn't go easily through the hydrophobic core of the membrane. So it's called facilitated when it has kind of a adapted channel, like a perfect channel for it to go through. For example, potassium. This would be example when potassium go through the membrane in an axon during the action potential. That would be an example of facilitated diffusion like this. Then 16. What is the mechanism of sodium-potassium pumps in neurons that generating the resting potential? And the important thing here to remember is that if you have a neuron, let's draw it here, and then let's just so here is my version of sodium potassium ATPase. No, sodium ATPase, sodium potassium cotransporter. So here we have the axon. Here we have the two membranes of an axon. Here is the axon. And then the important thing to remember is that when it's resting, you have the sodium outside and you have the potassium inside. That's not only true for axons, that's true for all cells. So A says, yeah, so let's look. So the question is, what is the mechanism of sodium potassium pumps in neurons that generate a resting potential by active transport? Okay. So maybe the yeah. So A, potassium from cytoplasm binds to the pump and stimulates its phosphorylation by ATP. So this pump does not transport potassium from the cytoplasm. The reason being, it transports potassium from the outside of the cell into it, because during the resting phase the potassium is inside. So that is wrong. B, phosphorylation of the pump causes its shapes to change in order to move sodium ions into the cytoplasm. No. This sodium is have a high concentration outside, so if it wanted to go inside, it would just do so with the facilitated diffusion or something. So this also is wrong according to the resting concentrations of sodium and potassium. C, potassium from inside the cell binds to the pump and causes the release of the phosphate group. Again, this is wrong because potassium, yeah, it's basically A. Because here it says from the inside of the cell and here it says from cytoplasm. Yeah, so they just repeated the wrong A in another way. Because it's yeah, the only different thing is if the which is when it's phosphorylated by ATP to transfer energy, or when the phosphate group releases. So D is the correct answer here. Sodium from the cytoplasm binds to the pump. Yes, that is correct. So because let's say there is a sodium here, it's not supposed to be there. So the sodium, you see here, it goes into here, and even two more needs to go here. So now it's filled with two more, and then when this is full with 3 sodiums, then ATP, which is an adenosine with 3-phosphate, it transfers one of them and becomes ADP, and then this kind of phosphate group here represents a lot of energy. So it has a lot of the chemists in your class, or maybe you yourself, if you are one of the chemist students, would say that this represents a lot of enthalpy, and this enthalpy, this energy, makes this protein transform so that these sodiums are released outside the cell. So D is correct. 17. What is characteristic of homologous chromosomes? So A, they have an identical DNA sequence. No, because You get one from mom, one from dad, and they don't have the same sequence. B. They are of the same length in karyograms, and yes, that's actually what you do in karyograms. You take chromosomes of the same length and put them together. Why are they the same length? Well, each chromosome has a set of loci. That's plural of locus. And the loci contain the genes. And even though genes can be different, the loci are exactly the same. They like a bookshelf. You can put books with different content, but the bookshelf is always the same size. And when you have many identical bookshelves, you get the same library. Yeah. So B is correct. And C, they form pairs in prokaryotes. Prokaryotes have only one circular DNA. D, they carry the same alleles. No, this is kind of the same as A, but it's still wrong. Because, for example, your mother could have the allele for green eyes and your father blue eyes. 18. What event occurs only in meiosis? A. Fusion of gametes to promote genetic variation. Well, meiosis happens before the fusion of gametes, so don't know what to say other than it must be wrong. B. Random separation of chromatids. This is a very hard, like this is very utspekulert, we say in Norwegian, like so in mitosis, let's say this is chromosome 1, and then you have two homologous chromosomes, one that came from mommy, one from daddy. It is actually random separation of the chromatids, but it doesn't matter because the chromatids have not undergone crossing over. So each homologous chromosomes have two identical sister chromatids, this one and this one. So even though it's random separation in mitosis, it doesn't matter. So this is wrong because it actually doesn't only occur in meiosis, it occurs in mitosis also. But we don't think about it because it doesn't matter, because the chromatids are similar. Oh, what I'm doing, stop, like this. Okay. Pretend you didn't see the queen. Okay. C, random separation of homologous chromosome. And of course that is correct. And this happens in meiosis 1. Because let's color one of them and say that's can't I color? Let's color this one and say it's red. So this is from mother, this is from father. So during meiosis 1, all the homologous chromosomes are lined up, and then these two can go. This can go to one cell, and then this one to the other, or vice versa. So it's arbitrary which of the daughter cells after meiosis 1 the different chromosomes go to. So for example, one daughter cell could get chronologically daddy mother daddy, and then the other one would get mother daddy mother, for example. So this is indeed correct. There is random separation of homologous chromosomes. Meiosis 1, I think it's anaphase. They are, yeah. Replication of chromosomes, no, that doesn't happen in either meiosis or mitosis. It happens in the synthesis phase of the cell cycle, so that is also wrong. 19. In which situations are alleles exchanged? And that is, so let's see here. A, during the separation of sister chromatids. No, it happens before that. In the transmission of linked genes, well, linked genes are transmitted together when there were no crossing over. So of course, I didn't say so, but exchange of alleles are crossing over. Crossing over. See, during fertilization, when sperm and egg chromosomes pair up. No, it happens before that. When chiasmata are formed between non-sister chromatids, and that's of course correct. So let's make a bigger line here, a thicker line here. So let's say we have one chromosome here. And then we can take a red one. So here we have here I managed to make two chiasmata. So these two, here is a chiasmata and here is a chiasmata, and here the genetic materials, the DNA, can switch places, so that you get this hybrid Frankenstein chromosomes. So like this, and then this goes like this, and then there will be red here and here, and then we get the red one. So that would be like this. And then the missing parts would of course come from the black one. So we see that we exchanged here. No, we exchanged here and we exchanged here. So D is correct. And these are called chiasmata. And what are non-sister chromatids? So this is one chromatid, and its sister was this one before the crossing over. So maybe I should do this here. So if I take this one, so a sister chromatid are, for example, this chromatid is a sister with this one, but if we take but this chromatid is a non-sister with this red one. Okay, so these two are example of non-sister chromatids. So we see that makes sense for D. 20. What is the effect of dominant alleles? They mask the effect of recessive alleles. Yes, they do. They become more frequent than recessive alleles in the population. Well, not on the genotype level. They might overshadow many recessive counterparts, but they don't actually become more frequent. You just see them more. They have a joint effect with recessive alleles when characteristics are codominant. No, I don't agree. Like, they always overshadow recessive alleles, but they could have a joint effect with another codominant allele. So I would say A only. One only, meaning A. The statement is about genetic control of cat coat color. And here they describe tabby cats with stripes and blah blah blah. But the interesting thing here, so usually in this exercise you would have to make a chi-square, no, not what's it called, Punnett square with two characteristics. So you would have to make, so the stripes are, the stripe thing are A or A. No, that's the stripe thing is M and M, and two-toned here is A and A. Okay, so stripes and tone. But the funny thing here is that the recessive allele A causes all coat hair to be black and so hides any pattern. So there are two ways to solve this. You can either make all the genotypes from two traits. So for example, let's say you make all the possible male phenotypes. So that would be any combination, and then the cats are heterozygous, so it would be MA, 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 and MA, like this, and then the same for the egg. Okay. And then you would cross them. So this would be M, M, A, and so on. And then maybe here you would get the M, M, A. And you would count how you would say, oh, this one is black. Because, look, A causes coat hair to be black and hides any pattern. So, yeah, this would be black, okay? But since this hides any pattern, so that means that AA quenches in a way. Quenches expression of the stripe. So even though M is dominant in the stripe formation, or if it's stripes or swirls or so, it doesn't matter, because if you have the recessive homozygous small a, it is black anyways, regardless if it's stripes or swirls. Swirls. So you can do it this way and use a lot of time, or you can just understand that the recessive homozygous is black no matter what happens with the stripe gene. So you could just set up this one, A, and voila. You see that 25% are black, and that is this one. Black cats are 25%, which means 4 out of 16, because 1 to 4 is the same. If you multiply with 4 up and down, you get 4 to 16. That is indeed C. '22. This is the cross that led to the discovery of non-Mendelian ratios in Morgan's experiments with Drosophila. That's the banana fly. And the thing here is that you have one chromosome that has maybe B plus here and maybe VG plus here. So one parent have only plus versions, and then another, and another chromosome had maybe only without plus, and then these homozygous were paired to make a hybrid. So all offspring has one chromosome from father, one from mother, and so the plus versions are situated on the, for example, the father chromosome, and the non-plus version are on the mother chromosome, and they are never together. Okay, but as we know, when these make offspring again, there can be a crossing over during meiosis. And so the only thing we have to look for here is in this notation, because this thing here is one chromosome, okay, and this thing is one chromosome, another chromosome, and so this notation is so easy, because one line, one chromosome. So you just have to look for one line which has one version of the gene which is non-plus with a plus, and we find it here. Let me see. We find it here. This is the culprit, because here we have a non-plus version and a plus version, And that means that crossing over must have occurred, meaning that, let's say, Vg plus went over. So this must have gone over to this one. So you get, you keep the B without plus, but you get the plus here, okay? So B is the correct alternative. 23. Which are characteristics of a species? The potential to interbreed, to produce fertile offspring, yes. And the formation of a population with members of the same species within a community. So it basically means that individuals of a species bunched together, I would say it's true. So 1 and 2. Let's see if that was correct. 3. Oh, yes, it was correct. 24. Statement about the role of some bacteria in ecosystems. What is the mode of nutrition in these bacteria? Okay, so these bacteria, they live on organic wastes, so dead material, and digest their molecules. So they don't use pure inorganic chemicals. They don't use sunlight and photosynthesis. So what is the mode of nutrition of these bacteria? So they are not autotrophs, because then they would use sunlight or maybe sulfur or something inorganic. They are consumers. Well, consumers, they kind of eat living producers often. So you wouldn't say they are consumers. They are saprotrophs. Yes, they are. They are detritivores. Well, detritivores are similar to saprotrophs, but detritivores are like wood lice. Have you seen those? And lots of, and they ingest the dead organic materials. That's more a detritivore. So C, saprotrophs, correct. Fungi also are considered saprotrophs. 25. The diagram represents a pyramid of energy. What level does the X letter represent? And of course, here, you know, you have the sunlight. So, so the red is absorbed often. The green is reflected. The blue light is absorbed. So, what level does the letter X represent? Well, not light. Primary consumers like sheep, no. The abiotic environment, no. I don't even know exactly what I mean by that. But producers, yes. So grass, for example, are producers, or algae in the marine ecosystem. And also, when you have a pyramid of energy, it's like every level is 10% of the previous, okay? But if it's a diagram or pyramid of nutrients or something, it can deviate from this principle or norm. How can mollusks and Plathiel Mintus be distinguished? And so mollusks, like a snail, it has an anus and it has a mouth. So it has a mouth and an anus, I think. I don't remember exactly where. Plathiel Mintus have kind of a feeding tube, but it doesn't actually have like an anus. I think it somehow have these tubes, and then it just removes waste directly from the tubes via diffusion over the skin or something. So this is a mollusk. Yeah, and the Plathiel Mintus also have these pigmented areas, which are something alike to eyes, but not that advanced. Plathiel Mintus. And then mollusks are unsegmented, but Plathiel Mintus are segmented. No, both are unsegmented. Mollusks have a mouth, yes, and an anus, yes, but Plathiel Mintus does not, and this is correct. Because even though Plathiel Mintus have a kind of a mouth-like thing, this tube, it does not have an anus. 27. Did we do 26? Yes. Which is the most efficient way for some desert plants to conserve water? And desert plants, they have done something really cool. They have just gotten rid of leaves, or maybe not got rid of them, but they have transferred the leaves to spines. And it's very good for the cactus to have a lot of spines because it protects it from all the animals in the desert that want the water. So let's look here. Which is the most efficient way for some desert plants to conserve water? So let's look at A. By having no leaves, so water evaporates from the green stem with less surface. I think this is the correct one. Because the leaves would increase surface area, you would need more stomata, and that would be a problem. B. By loading organic compounds in the phloem of the roots. I'm not sure how that would help anything. By accumulating salt within the tissues, so water is retained by osmosis. Well, there is no water around the cactus. It's completely dry, so that wouldn't help. By growing long hair on the surface, so air moisture is absorbed at night. And here they are referring to old man in the desert from Mexico. So this old man in the desert, it has a lot of hair around it, but it's actually not for capturing moisture during the night. It's for kind of stilling the air that flows over the open stomata during the day when it has photosynthesis. Remember, plants need open stomata to capture the carbon dioxide. And if you can get the light, have the stomata open, but not let the water vapor drift away during hot desert winds, that is really good. So this D has some truths in it, but it's not correct in the end. 28. What allows most plants to continue producing more roots, leaves, or stems throughout their life? And that's, of course, meristems. Meristems are kind of the tissue in plants that can divide and produce a new plant. Like, very unlike, for example, animals. Like, if animals were like plants, we would have kind of pluripotent stem cell tissue that if we cut off a finger and put it in a petri dish, we could grow a new human or something. Plants can do that. It's fascinating. 29. How do most angiospermophyta improve sexual reproductive success? And of course, angiospermophyta are the flowering plants. And they have made this what's called cooperation with insects and other pollinators, like hummingbirds. So it's C. And mutualistic means both gets benefit. Both parties benefits. 30. To which domain does Carcarias, a shark, belong? And of course, that's a fish, and fish is an animal, and animals are eukaryotes. So remember, the domains are bacteria, eukarya, and I forgot what their name was. Archaea. The archaea. 31. One picture shows skeletons of a frog and another of a domestic rabbit. What is the evolutionary relationship between X and Y? And we see that it's kind of the same leg here, because here you have the hip bone, and then you have the first bone. It's called a femur or something. I don't remember exactly. Here, the hip bone, I think this is still the hip bone, so this is the same bone. And so the tetrapod was kind of the first animal we see that walked on four legs. And so, yeah, this was a leg from the common ancestor, the tetrapod. So what you say then is that not only are they alike, they are homologous. What we mean by that is that it's the same structure. It was the same structure in the ancestor. So this question is poor because it is a little bit confusing for a very strong student. How can this be? How can the strong student miss the correct answer here? Well, bats and birds are analogous when it comes to wings. Why is that? Well, the ancestor of bats was a dog-like creature which had 4 paws used for walking. And the ancestor of birds was a dinosaur maybe catching insects with the 4 paws or something. But the thing is, the common ancestor of bats and birds did not have wings. So if you ask about wings, they are analogous in bats and birds. But if you ask about 4 paws, they are homologous because the ancestor did have 4 paws. And you see the problem here, because both frogs and rabbits, they use their backbones for jumping. And so a strong student could be confused there and think, the common ancestor of frogs and rabbits didn't jump, so he would maybe say analogous, but that would be wrong, because we are not talking about jumping legs. We are just talking about femur, one of the bones. 32. How can species of bacteria evolve to be resistant to antibiotics? And of course, first of all, you need a variation, or you need kind of resistance gene or something in at least one bacteria. And then number 3, that bacteria needs to survive. So an incomplete dose would be very beneficial. So if you have a lot of bacteria here, so it says that this one bacteria has resistance, and then if you use antibiotics but too low dose, then this bacteria will survive and it will multiply and form a new colony. And this is what actually evolution means, that you have a lasting change in the genetic makeup of a population. But if you used a very strong dose, high dose, maybe there was no bacteria that survived at all, and then even though there was a potential in this bacteria, if it died because the dose was just too high, it's okay. So to get evolution of antibiotics resistance, you need both 1 and 2, 1 and 3, meaning C. 33. What happens to starch in the small intestine? Well, starch is a polymer of glucose. So it says endopeptidase secreted by the liver, blah blah blah. Well, endopeptidase, it breaks down proteins, not carbs, so it's wrong. B. Contraction of intestinal muscle mixes starch with enzymes. Okay, looks promising. To accelerate its conversions into amylose. And what is amylose? It's actually a component of starch. Like, I think amylose is this one, and then amylopectin is the branched one. I'm not sure, but anyways, like you don't digest starch to like just transform which type of polypeptide it is. C, so B is wrong. C, glycogen secreted by the pancreas. So, okay, glycogen is the animal storage carb, and it's a polymer, and it's never secreted, like only monomers are secreted ever, so no. And the pancreas secretes hormones and stuff, like if the liver can secrete glucose but not the pancreas. Okay, so all kinds of wrong things going on with C. So of course the correct one is D. Amylase, that's an enzyme. Look at the ase ending. Secreted by the pancreas. Yes, pancreas releases a lot of digestive enzymes. Digest starch. Yes, to enable absorption by the villi. So the villi creates the large surface of the small intestine, but they can only absorb monomers, and the amylase enzyme helps with that. So yes, that's correct. 34. In which process is surface area of key importance for humans? Reabsorption of glucose in the proximal convoluted tubule, and that is indeed correct. But just let us look at the other ones. Release of surfactants by type 1 pneumocytes. Well, it's type 2 pneumocytes that release the surfactants, but anyways, surface is only important for type 1, which are where the gas exchange occurs, so this is all ways of wrong. C. Display of antibodies by red blood cells. Well, red blood cells are actually not involved in the immune system in that way. And anyways, you don't display antibodies, you display antigens. D. Secretion of enzymes by villi of the small intestine. Well, villi, they absorb nutrients, they don't secrete enzymes. They are not glands, so these are so wrong. So this one could easily be, even if you don't read much about the kidney, you could see that A was the correct by using elimination. So what happens? So the Bowman's capsule empty. So you have the glomerulus inside here, glomerum, and here you have a filtration of the blood, and the ultrafiltrate goes into the proximal convoluted tubule. And what happens here is that if you got some glucose through, you want to keep it. So it's kind of pumped. I think it's a sodium glucosim port, meaning it uses the gradient of the sodium to transport the glucose or something. And it's put into the peritubular capillaries. So the glucose is transported into the peritubular capillaries. Fun fact, the kidneys is one of just a few tissues where you have 2 capillary beds. One here in the glomerulus and another here between the tubules. 35. Which discovery was an indication that the heart pumps blood to the body through arteries? And A is wrong because it's a closed loop, so you can't have different amounts of blood. So A is wrong. B, blood could easily be pushed up a limb vein. I'm not sure how you would define up. Like in veins and stuff, you have the proximal and distal part. I don't know what would be up. See, the observation that there were pores between the right and left atria. Again, we have a closed loop. I think reptiles have openings between them, maybe. Like at least some more primitive animals have it, but we don't. That's wrong. D, the heart swelled up when the arteries were tied in an animal experiment. And yes, of course, if you tie the artery and all the valves in the heart is one way, it would swell up. So yes, this sounds correct. 36. Which is a valid comparison between arteries and veins? And here we have to remember the large and the small circulation. At least that's what are called in Norwegian. In Norwegian. So the large circulation goes to the body tissues, and so we have the heart here. But the small circulation goes to the lungs. Okay. And the thing is that deoxygenated blood comes from the body and are then transported through arteries to the lungs, okay? And veins from the lungs carry the oxygenated blood. But this is opposite in the large circulation, because there it's oxygenated blood going to the body tissues and then deoxygenated blood returns, okay? So A is wrong because even though some arteries carry oxygenated blood, not all of them do. And same with veins. Same problem with veins. Some veins, those from the lung, do carry oxygenated blood. So B is wrong because so A is not a valid comparison. B, return blood to the atria of the heart. Well, yes, but only in the small circulation from the lungs. Yeah, so this is also wrong because in the large circulation the arteries do not return blood to the atria. It actually goes from the ventricle to the tissues they supply. See, the blood flows at high pressure. Yes, that is correct. Maybe we have the correct one here. Blood flows at low pressure in veins. Yes, so it seems C is the correct one. 37. Which statement is true for the antibiotic penicillin? And it says, Watson and Crick developed the usage of penicillin. Well, they actually discerned or found the structure of DNA. So A is wrong. B, penicillin blocks processes unique to eukaryotic cells. No, actually, penicillin is kind of an antibiotic, meaning it affects bacteria, it disrupts bacterial metabolism, so that's wrong. C, viruses lack metabolism, and penicillin has no effect on them, and yes, that is true. Like on the virus itself, penicillin has no effect. Maybe if a bacteriophage, which is a bacteria virus, infects a bacteria, and then it could be subject to antibiotic action, but that would be a special case. So C is the correct alternative here. 38. Which process is part of the mechanism that controls muscle contraction? Troponin enables. This 38. We just have to draw it up. Okay, so this is an actin coil. They go like this, and then maybe there are some actins going on behind here, but let us not focus on them. And then you have tropomyosin, which covers all the binding sites. Okay. And then in between those again, you have some troponin anchoring points, shall we call it that? And the thing is that this tropomyosin, it is, it's covering the attachment points to the myosin heads. So the myosin heads are like this. Attached to the myosin fiber, and it wants to kind of attach here. No, let's say here. But it can't because it's covered by the red line. So if calcium, so let's say this is calcium. If it attaches to the troponin here, let's make it a little bit smaller so you can see that it fits inside the troponin. It doesn't work. I need to make a smaller one. Let's make it this small. No, it's really small. No, that's too small. Let's make it this small. Yeah, perfect. So this is calcium, okay? This black one is calcium, and it attaches to troponin, and this makes actually troponin and calcium to move away. So it kind of displaces them in a way. But what happens then is that the whole thing moves away. So this one moves, this one moves, this must move. Like it's dragged away so that the binding sites for myosin, the myosin heads are uncovered. And so then this can drag like a dinosaur head, and then it jumps to this one, and this climb of the myosin hand and on the no uncovered binding sites on the actin makes this fiber kind of move alongside and actually reduces the muscle fiber. So let's see if some of these alternatives capture this. So calcium frees actin filaments, no. Tropomyosin attaches to calcium, no. Myosin heads attach to troponin and tropomyosin pulls on no. Troponin enables actin heads, no, they are myosin heads. Let me see here. Calcium freeze. It seems like D is indeed the correct one, because it says that calcium frees actin filaments. Yes, this is correct, because calcium binds to troponin, this red blob, and displaces the whole thing. And since the troponin is attached to the tropomyosin, okay, so tropomyosin, troponin, and this is calcium. For myosin heads, yes, this is correct. This is indeed a myosin head. To attach, yes, that is also correct, because the myosin head attaches like this, and reduces the muscle fiber. Yes, very good. So we found out that 38D was the correct one. 39. Which sequence of events leads to the production of antibodies? And I can just say immediately that A is the correct one, because you have the non-specific immune system with phagocytes, big blobby cells that eat stuff, and one of those are called dendrites. Just mark them with D. And dendrites have the good habit, the nice habit of displaying things it ate. So let's say this red thing is, let's say, bacteria protein, bacteria surface protein, or maybe coronavirus surface protein, okay? So then it's called an antigen, like it's something used by the immune system to recognize pathogens, and then we just call it an antigen, even though it's actually a peptide fragment or something. And then if you have a T cell, a T helper cell, it kind of recognizes this, and woo, we have some baddies here. And then it releases cytokines that again activates beta cells, just let me call them B. And if one of those can actually catch coronavirus with that antigen, it becomes very stimulated, starts dividing like crazy, mitosis, and forms many plasma cells that produces antibodies, which fits this corona thingy, antigen. The antibody fits the antigen, but it also makes some memory cells, and those goes into the storage in the bone marrow. And if kind of you are infected again by corona in many years, then this will very soon multiply, so you get a very rapid response of the specific immune system. So we see that this is correct. Yes. Activate by cytokines. Yes. And B lymphocytes are selected, and if they can make the right antibody, they can make a clone of memory cells and a clone of plasma cells, which makes antibodies. Yes, everything is correct. 40. The image shows a section of a seminiferous tubule. This is the last question, and I think that so we are missing the image here, but A is the correct. So if I remember correctly, so if you have the scrotum, yeah, you have seminiferous tubules here inside, and it's in the scrotum to get the temperature lower than 37 degrees, because meiosis doesn't like 37 degrees. T less than 37. And this seminiferous tubule, let's make it here, June, and then you have epithelial cells here. Epithelial cells, and then maybe you have a Sertoli cell here. Sertoli cell, that's a food station. And so spermatocytes that are finished with everything, both meiosis 1 and 2, they are feeding here and growing and maturing, or maybe just waiting. So if they have meiosis here, mitosis here, when you get closer to them, to the thing in the middle of the tube, like the open, what's called the tube center, they become more and more mature. So I would guess like meiosis 1 are going on here. So here is meiosis 1, and maybe these cells are meiosis 2, because now they are soon finished, and maybe these have like the black thing in the front, and these are ready to go. They just need some extra food. So I would guess that the X would be somewhere around here, but I'm not sure since I can't see the image. Okay, so this took quite a while, 1 hour and 22 minutes, but maybe you got something out of it, like you saw the way I was thinking, excluding some alternatives and so on. I think the exams are in May, maybe 19th of May or something, don't quote me on it. But I think if you are biology students in 2021 spring, then you have some days, and I plan to maybe release maybe 1 or 2 videos a week until the biology exams. So click the subscribe button and maybe like this video, and then maybe I will make more videos. So see you.